Hi, I am Kevin Roelof. I'm the Associate Director of Strain Process Development at Novome Biotechnologies, and I'll be attending the 8th Microbiome Movement Drug Development Conference. Very excited.
Perfect. Well, very much looking forward to having you speak at the conference in June. And, with that said, I just wanted to go into our first question, which, there's going to be over 250 world leading microbiome professionals united at the meeting. What is the importance to you in attending these meetings?
Yeah, great question. For me, this is really about building the community. Um, it's been a hard time in drug development in the LBP space, and now more than ever, we need to come together to share ideas about what works and what are the challenges we're facing and how we can overcome this together. I think I also really appreciate that the Microbiome Movement Drug Development Conference brings together a lot of the industry that's really gotten more diverse over the years. So there's a incredible range of types of products, from the donor-derived fecal transplants to the consortia products of individual strains, or even, as Novome is doing, the genetically engineering of some of these strains to give entirely new capabilities. And coupled with all these different ways of solving the problem are the vast number of ways that microbes can help humans. And so I'm really inspired by the opportunity to kind of make a new type of medicine that can help people. And it's really interesting to see all the other people who are trying to do this and the ways that they're um, bringing their technology to bear on important problems for patients.
Well, you mentioned novel manufacturing tools and technologies. So that brings me on to my second question. As these tools and technologies are being developed to support scale up and more sophisticated manufacturing of live bacterial products, what excites you the most and why?
I've found this has been really interesting as I've gotten more and more into how you manufacture these products. And I think, a lot of times I've looked into the past to understand what the future will be. And traditionally, in the world of therapeutics, microbes have been the means to the end of producing proteins or nucleic acids. And this time around, in this field of medicine, the live biotherapeutics, the cells are the products themselves. So that creates significant more complexity, but it also means that we have an expanded toolkit to really take on more powerful approaches that incorporate some of the insights from some of the most rapidly developing fields in molecular microbiology, human microecology, and synthetic biology. And I think that really brings a lot of the opportunities from academia, starting to make them into products that can actually help people. And, so there are the methods about how to produce and stabilize and characterize and control live anaerobic bacterial populations. A lot of these have been developed outside of the pharmaceutical industry, in academia, agriculture, food processing. So the challenge we face today is to understand what the solutions were found in other industries and figure out how to translate them into the pharmaceutical world, where we need to have an increased level of rigor and control around these manufacturing so that we meet high quality standards that ensure patient safety and then efficacy of these products. Um, and I think these quality standards, a lot of times people are looking for clear understanding of what they are. But I think it's rather an exciting time where the regulatory agencies have recognized that there's a new opportunity here and they don't have all the answers. And they're asking for sponsors to come up with risk-based methods and to understand bringing new technologies in, such as next generation sequencing, which kind of built the whole microbiome field in the first place, or microencapsulation, or modernized methods of anaerobic culture. We can't all be screwing Hungate tubes together forever. We got to figure out how to keep the oxygen out. Anyway, so harmonizing all these elements into CGMP standards is a big task, but it's kind of exciting, and I think there's an era of collaboration in the field.
What's the long term value in attending these definitive microbiome movement meetings?
Yeah, so I mentioned I was looking into the past to kind of understand the future of microbiome therapeutics. And particularly, I was interested in understanding kind of how monoclonal antibody production occurred, because many of the manufacturing processes are somewhat similar. Um, and one thing I came across was a talk describing how the maturation of CMC for monoclonal antibodies, um, first there were some pioneers, but it was an extraordinarily expensive product. And there were a lot of patients who needed it who just couldn't access it, whether it was because they didn't have a cold chain that they needed things to be transported in -80 degrees C freezers, or it was just too expensive. Patients couldn't afford it. And one of the key lessons from this talk about monoclonal antibodies was how the organizations that were potentially competitors with each other started cooperating to share understanding about methods, to standardize some of the upstream and downstream processing, to share capacity within large manufacturing plants that often cost billions of dollars to set up. And if those sit idle, it's really, increasing the cost of providing these drugs to patients. And so it started to be individual people within these organizations coming together and making a network of partners instead of competitors that could share and start to learn from each other and share resources that really started bringing these drugs in large scale to patients and modernized some of the therapeutic world. I think the live biotherapeutic field is in a similar area where we're just still figuring out what is working, but we're starting to standardize this a little bit. And there's been large investments in the manufacturing, as we'll see from some of the other talks in this microbiome movement conference. But I believe here now is the time to build the relationships among the various people working in this field, such that we can start to cooperate instead of just be competitors with each other. And I'm really inspired by the opportunity of this conference to provide that opportunity.
Thanks, Kevin. Really looking forward to having you on board and seeing you in June at the meeting.
Yeah, thanks, Lily. It's been a pleasure to talk to you.